Cheers and happy Saturday to you all. Now that we've talked about mutation and how to recognize the different ways in which a mutation can occur and the different downstream impacts of those mutations, I want to talk today about how we detect mutants. That is, if we want to actually go on a mutant hunt, how do we find one? How do we capture it? How do we differentiate it from all the other wild type cells around it? So there are different ways depending on what kind of mutant you're trying to hunt. So for example, if we are working with Micrococcus luteus, now remember when you looked at Micrococcus luteus on the plates, it was bright yellow. If you wanted to find an albino mutant of Micrococcus luteus, you could do that by simply observing it. You would look for the one anomaly on the plate that rather than being bright yellow was white in color. And then you would be able to verify, right, that it was in fact also Micrococcus luteus, but it just lacked that pigment. So this is one example. By observation, we could detect a Micrococcus luteus mutant that was albino by observation. Likewise, if we were looking for a Pseudomonas aeruginosa mutant that didn't produce piocyanin, the blue-green water-soluble pigment, we could do that also by observation. It would require that we put it under a little bit of stress and that we allow it to age a bit, but we could also look for something like that via observation. We can also talk about our superhero mutants or our X-Men mutants, right? Not all mutation is deleterious. Some forms of mutation endow superhero powers. So with our superhero mutants, we could put them in an environment where they would live and everything else would die. For example, if there were no rain, well, Storm would live because she could conjure up her own storm and she would be drinking when everyone else was dying. It kind of conjures a gruesome image, but in fact that is the way that we go about selecting for superhero mutants in the lab. Remember when we were looking for the streptomycin resistant E. coli mutant? The way that we did that was to add streptomycin to the plate, and while everything else was dying, the streptomycin resistant mutants were living. So we could find it by killing everything else and only the superhero would live. This is a form of selection called direct selection, but it only works if it's a superhero mutant. If instead we have a mutant that lacks a capacity, things get a lot harder. So in a moment, we'll talk about how we would go about selecting for a mutant that lacks an ability, but before we do that, we need some lingo. So the lingo around that is prototroph versus auxotroph. An auxotroph is a mutant that has lost its ability to synthesize some sort of growth factor, and therefore it requires that we put that growth factor into the medium, otherwise it will die. So a good example of an auxotrophic mutant would be something that had lost the ability to biosynthesize, say for example, tryptophan. Remember looking at the trip operon in our activities last time. So if something has lost the ability to biosynthesize tryptophan, it would require that tryptophan was always provided in the culture medium. Versus a prototrophic or wild type organism that can synthesize its own tryptophan. It has that trip operon, and when tryptophan is not around, it just simply forms the enzymes needed to biosynthesize its own tryptophan. So we could say that a prototroph requires the same nutrients as a naturally occurring member of its species. If a naturally occurring member of its species can make tryptophan, then it can also make tryptophan. It can grow on the most minimal media that a natural occurring member of its species could. So if it can biosynthesize tryptophan and other metabolites, it could grow, say, on a GSA, where only glucose and salts were provided, that it could build everything from that. It has all the functional biosynthetic pathways for the synthesis of growth factors. So what if we wanted to find an auxotroph? What if the mutant that we were hunting is the one that had lost the ability to biosynthesize something? This hunt gets really hard because unlike direct selection, where we can simply look for it based on an ability, an auxotroph has a lack of an ability. That's really a lot harder to find. Let's look at an example. An E. coli strain that has lost the ability to synthesize lysine. We have some terminology that goes with this, so we get a little sexy-minded. When we write the genotype of this, it's a lowercase l and italicized. We say that this lys minus genotype is associated with this particular auxotroph. So sometimes we also say delta lys, so delta meaning it lacks that. So two different ways to indicate the genotype. The phenotype is indicated similarly, only it's not italicized and it's an uppercase L. This is a common differentiation between genotype and phenotype to use that form of distinguishing. So let's think about this. If we had an E. coli strain that had lost the ability to synthesize lysine, what we know is that it would grow fine if we put it on a rich, undefined medium. Like if we put it on a TSA plate, it would probably be okay because lysine is already provided in there. But if we put it on a GSA plate, or a GSA plate, we wouldn't see it growing very well because lysine isn't provided on that plate. So this makes things hard because if you put it on a medium that it will grow on, so will the wild type cells. So everything will grow. So say we're looking for this lys minus mutant, we put it on something that will support its growth, well, everything else grows there too. So now, by the flip side, if we put it on a medium that it won't grow on, the only thing we'll see is its absence. So it won't grow on a GSA or a medium without lysine. So everything else will grow there, it just won't be there. So there's just no way to directly select for this sort of mutant. So how do we do this? Well, we get tricky. We do indirect selection. So in order to hunt and capture an auxotrophic mutant, we have to actually look for its absence. But how do we look for its absence and still grab it and still get a colony of it? Well, here's how we go about it. We do something called replica plating. With replica plating, we have a wood, or in our lab, it's a synthetic block. And the synthetic block has rivets in it where you can screw in a ring. And you put a piece of sterile velvet over the top of the synthetic block, and then you screw in the ring to hold the velvet tight. And then you take that sterile velvet and you press it down onto a master plate that has both the prototrophs and the auxotrophs. So essentially you're printmaking. You're literally making a print onto this sterile velvet. Why velvet? Well, because velvet's like 100 million little inoculating needles that are all picking up the colonies. So now on this sterile velvet, we have a print of the master plate. We can now take it and put it onto a control plate, a TSA, that we know will support the growth of both the auxotroph and the prototroph. We then put it onto a GSA, which we know will only support the growth of the prototrophs. The control is everything, because on the control, the auxotroph will grow. But look at it, it's gone on the GSA. So we can now find this little guy, we can pick it up off the TSA and say, you little one were the one that didn't grow on the GSA, but you did grow on the TSA. Looks like you lack the ability to biosynthesize something. Of course, with this method, we don't know what it lacks the ability to biosynthesize. That sounds like a job for a grad student, right? Now, you can make a working stock and a freeze down of this presumed auxotroph, and you could take it and try to streak it on a GSA containing lysine, a GSA containing tryptophan, a GSA containing, whatever growth factor it is that you think it can't synthesize, and finally you'd find something that would promote the growth. And then you could say, hey, I found, a gly minus mutant, that was what promoted the growth. Let's take a moment, and unfortunately because I'm socially isolating in my home instead of in the lab, I can't show you replica plating, so I'm going to post a video that shows a nice example of replica plating.